Hi there, and welcome to a video outlining some other types of inheritance outside of just that straightforward Mendelian pattern. So what we have learned sort of observing the inheritance of traits over the years, that it's very rarely simple. With genetics, there's multiple genes sometimes in play, or a gene can have multiple effects or not be expressed in the way that you might think. So what I'm going to do is kind of use a couple of very straightforward alleles and then kind of compare what the patterns would be like. So if we were going to use, let's say, flower color, that would be our gene. And we made, we'll make R the red allele. And then we'll make CW the white allele. And then we'll kind of compare some of the effects that you have. So you could set up a Punnett square with two heterozygous individuals. If you want to make your flowers male and female, that's certainly your prerogative there. Okay, so we'll make them both heterozygotes just so we can get some different combinations going on here. Notice I haven't defined dominant or recessive here. Okay, so I'll put some different combinations that are here. This is not a sex-linked example, so we don't want to kind of throw that into the mix just yet. And again, just by convention, we typically choose to write one of the letters first. So I chose R first, just so I can keep track of which ones are the same. Okay, so what I'm really interested in here are the heterozygotes. So I'm interested in the two different combinations that are here for most of my examples. So in a continuous variation, any of these examples that I've given you will, so it's one gene, sorry, let me back that up. That's one phenotype it's influenced by multiple genes. So we often look at that example with height, skin color. There's kind of a range here that you can see, and typically there's different genes that are involved there. So I only have a one gene example. So the way I would compare this to, plug in this flower color example, is that this wouldn't be the only set of genes that would influence flower color. And we in fact do see that quite often, where it seems like there's one gene that influences a particular trait, but there's actually other genes that need to express themselves in a particular way in order to get that trait to be seen as a phenotype. So you would have, instead of just the CC combination, maybe you would also have a set of P genes that has to do with petal, petal protein or petal response to sunlight. So there would be other genes that would influence that. Now in a pleiotropic setting, you have one gene, so it's kind of the reverse in some ways. One gene has multiple phenotypes or multiple expressions. So we see this in disease genes, like something like sickle cell, where we know it's one set of genes, but we see a variety of symptoms. Same thing with cystic fibrosis. So we know it's one gene, but we see a variety of symptoms. So we're calling all those different symptoms or impacts phenotypes. So if we were to look at this flower color again, we could say that if you were to have any of these combinations, it might influence not only the color, but it influences how early they open in the year, or what the size of the petals or the number of the petals are. So it's multiple effects. So maybe you would always see that the red petals are a smaller size and the white petals are the largest size, and then maybe this is somewhere in between. So it has some impact on other phenotypes as well. So an incomplete dominance, and now I'm going to look at my heterozygotes. I'm going to focus in on those that I circled a little bit. Incomplete dominance is where you would get a blend, if you will. So now your It wouldn't matter if it's a homozygote, what it's right, because it's the red blended with the red. But if you had the CR and CW combination, instead of getting red or white, you get a blend, so you get pink. That's incomplete dominance. That's different than codominance, because in that case, you have two distinct phenotypes. So I wouldn't get pink. What I might get is something like red and white polka dots. So I would see both of them there, or red and white stripes on my petals. So I would see very distinctly. And one of the places we see this a lot in humans is in blood type, because blood type, we have one codominant arrangement. We have the type AB. Sometimes people are all type A alleles, so then they have type A, or they're all type B. But if they have one allele type, if they have an A type and a B type, they don't just, one doesn't dominate over the other, both proteins are present, so that's a codominant. So remember, those are two distinct presentations, so you would see both of the phenotypes expressed, where in incomplete you see a blend, they're kind of mixed together. Now, environmental effects, so this impacts expression. And you see this in animals a lot in coat color, because they'll have, they have the genes, right, for a certain fur color, but they might have certain patches, maybe around the ears or the feet, where they have distinct colors, like, you know, darker feet or lighter feet, or around the ears or the muzzle. So it impacts expression, and in that case, a lot of times it has to do with the temperature of the body. And so the different temperatures in different parts can cause different effects. So for example, let's say we go back to any of our expressions here, but let's look at our heterozygote. Let's say that this particular seed germinates in a colder time, like in the fall, for example, as it's getting cold. The temperature, as it's getting colder, may somehow diminish the red expression. And so you see the white expression in the fall, even though we know it's a heterozygote. However, if the same plant germinates in the spring with the same genotype that's here, then the red does dominate, because in warmer temperatures the red is expressed. So the environmental effects can do that. And you know, this can get really kind of intermixed in terms of what the effects are, because we can go back to something like height, where we have on our continuous variation scale, it's influenced by multiple genes. Well, there's also environmental effects, such as the nutrition, right? So there can be different things. It could be thyroid function, right, that, you know, influences growth and things like that. So those can also influence, kind of cross-influence each other. So when we look at true Mendelian inheritance, we don't see that a lot, even though initially that was kind of thought it was kind of dominant or recessive. We know that there's sometimes multiple genes affecting a certain trait, or one gene that has multiple traits or multiple different phenotypes that are shown. And I mentioned some diseases where you can see multiple symptoms, right? Most diseases have many symptoms. The incomplete dominance, where you have a blend of the two, and one of the classic color, classic is the colors that are used for that, because you can see that nice blend of the two that's there. The codominance, where you can have not a blend, but they're both equally expressed, so one doesn't dominate the other. And then the environmental effects, and so that's probably influencing a lot of the expressions of these different traits that are here. So that's just kind of just a quick and short overview of some of the different patterns that you can see that are out there. Note that the Punnett square and the legend piece, those are the same. So again, I always go back to that's really key to kind of keep track of those traits if you're setting those up, whether it's sex-linked or on the autosomal chromosomes, the other chromosomes, but then knowing what the different traits are. So what I would do here, like if this were, for example, a codominant, I would add to my legend and I would write, oh, if they get this combination, they get, I put the red and white polka dots or something. So really having that key is going to be key to start to interpret those. So that's just an example of how you might use that. I thank you very much. This is Corey Andres at Central New Mexico Community College using CNM Glass. Please contact me with any questions that